enjoy the meeting.
Thank you very much. Good morning, ladies and gentlemen, distinguished speakers and guests. Yes, I would like to welcome you on the second day of our CETEF'22 on behalf of the Polish Chamber of Commerce for High Technology. Today, we start with the common session for two thematic conferences. One thematic conference is Emerging Biomedical Technologies for Diagnosis and Therapy. And here, the chairman of this conference is Professor Lucyna Woźniak, the Vice Rector of Medical University in Łódź. And the second conference is about breakthrough in Medtech. And here the chairman is Professor Robert Iskander. Just welcomed you from Wrocław University of Science and Technology. After this common session, we will divide into two groups. One group, the conference Breakthrough in Medtech, will stay still in this room, and the other group, Emerging Biomedical Technologies, will move into the other room. So I wish you fruitful discussions, and let us have a good day today. Thank you very much. And the chairman for this common part is Professor Lucyna Woźniak. The floor is yours, Lucyna.
Technical University, some technical problems are too difficult for me. I want to welcome all of you very sincerely, and I want to first of all, I want to welcome our eminent guests and speakers who decided to join us today during the session. I want to welcome everybody among the audience, because it is a great pleasure to meet after pandemic break and just to meet and talk and start new relation and improve and enhance before pandemic relations. I want to welcome also quite a big group of participants participating online, observing our session online, who could not come because of many reasons to join us here in person. So I hope that you will enjoy the session and you will enjoy the presentations and the discussions. We invited fantastic speakers and fantastic panelists. And also, I want to say that the program was constructed with strong help and active participation of several institutions, including our business partners, including universities. All the details you will find in our program, so I don't want to repeat it to shorten the time. So we will start. Unfortunately, one of our speakers could not come. It is Professor Abbas Alavi, professor in radiology and neurology and director of the research education in the Department of Radiology at the University of Pennsylvania. Professor Alavi was so kind and he wanted to join us online. However, because of some problems, we could not do it directly. Therefore, he recorded his presentation, and I hope to present it probably at the end of the session, not to ruin the discussion before. So let me invite these honorary chairs. And there is one armchair, I don't know why, maybe for elderly people. They have more convenient facilities. However, I want to invite in the alphabetical order because of the meaning of our guests and because of mixture of ecosystems they come and different disciplines they represent. I don't want to put a very specific order. We think that this mixture will create a very interesting discussion after short presentations. Our guests and panelists were invited to prepare short presentations, but it's not necessary. I hope the discussion will be something that is the most important during such meetings. So I want to invite Professor Adam Liebert from the Polish Academy of Sciences, the corresponding member of the Polish Academy of Sciences, director of the Knowledge Institute of Biocybernetics and Biomedical Engineering of Polish Academy of Sciences. I want to join us by Professor Michael Schroeder, professor in bioinformatics of the Technical University of Dresden, who is interested and probably will talk about machine learning algorithm, about applications to the evaluation of structure and sequencing data to improve diagnostics and treatment of infectious diseases and cancer. I want to invite Professor Olga Stepankova, representing Czech Institute of Informatics, Robotics, and Cybernetics. She's a member of the board and head of Department of Biomedical Engineering and Associative Technologies and deputy head of Department of Cognitive Systems and Neurosciences. I want to invite Professor Martin Różanek, who is associate professor at the Czech Technological University in Prague. So we have two representatives of the Czech community, who is vice president of the Czech Society for Biomedical Engineering and Medical Informatics, and Professor Jak Vilo, representing University of Tartu, member of the Estonian Academy of Sciences, full member of the Academy, and representing institute, being head of Institute of Computer Science at the University of Tartu. Professor Jak Vilo is involved in several initiatives in Estonia to build to create Estonian biobank based upon public-private partnerships. So very interesting and unique experience. I think he will also talk about this. And at least, but not at last, I want to invite Professor Roland Vogelmuth, who is president of the Swiss Corporation Committee of Biotechnology, but also professor at my alma mater, Technical University of Łódź. So Professor Roland Vogelmuth will represent a dual activity and real internationalization pathways in our research, which we are involved all. And having this fantastic panelists and in our mind, a presentation of Professor Alavi, I would like to invite Professor Adam Liebert to start with his presentation, which is titled Frontiers and Challenges in Biomedical Engineering, and I think will be a good introduction to the discussion. And I hope that Professor Liebert will take care of the panel discussion afterwards. Thank you very much.
Thank you so much. May I move this? Doesn't work. Really? I represent the Institute of Biocybernetics and Biomedical Engineering of the Polish Academy of Sciences. I just finished my duties as director of this institute in last month. Actually, I wanted to present you my vision of the mission of biomedical engineering. We'll show you a few examples how the biomedical engineering solved already big problems of medical community. I will make a short review of various projects which are very close to commercialization phase from my institute, but also from other institutions in Poland. I hope we'll have a chance to discuss the challenges which are present in everyday research of biomedical engineers. Depending on timing, I wanted also to discuss with you what are the unmet clinical needs where the biomedical engineers still have something to do. Claire Elwell from University College London said biomedical engineering is an area where we address the unmet clinical needs with innovative engineering solutions. The core of what we do are these unmet clinical needs, and these are still big challenges of the medicine. What we really do in biomedical engineering, we combine fundamental sciences like mathematics, chemistry, physics with various subdisciplines of technical sciences in order to solve problems which we know from biology, physiology, and especially in medicine. The research is highly interdisciplinary, application-oriented, needs close collaboration with life sciences experts, and interdisciplinarity is a core of many discussions of science today. We know that the impact of the research grows with interdisciplinarity. This is one of such presentations. But I wanted to stress that the connections between clinical medicine and engineering is still quite missing. So there is huge space of opportunities for us. And we solve these problems in various subdisciplines, including very classical like biomeasurements, biomechanics, biomaterials, but also emerging new subdisciplines like point-of-care diagnostic systems, nanomedicine technologies, biorobotics, etc. Which problems were already solved? Could you imagine therapeutic procedures in such medical theaters without all this equipment? Most of these treatments would be just impossible, and patients just die without this involvement of instrumentation. Biomedical engineers converted very primitive concepts into things which are portable, transportable, easy to use on the spot, in ambulance, and so on. Biomedical engineers converted the concepts known from physics, very basic concepts of imaging, from very primitive scanners into things of products of very high resolution and really easy to use for medical doctors in everyday practice. And this is maybe important for all decision makers to understand. This is just biomedical engineering. What we did, we converted this bulky instrumentation into something what is just a clamp on the finger, which saved plenty of lives during the pandemic. People have these instruments at home, yeah, and use them every day with advice of the medical doctors. So let me make a very brief review. I will not focus on any of these projects, just to give you some taste of what happens in the laboratories what the biomedical engineers have on their desks, and what they can offer at the moment as some surrogates of the future commercialization process. In my institute, Professor Darowski developed a computer model of respiratory and cardiovascular systems, but this is actually such hybrid system where we combine the software part with the hardware. So you can attach to this system, we call this artificial patient, various physical devices supporting lungs and heart, and this can be used for testing of devices of this kind. Professor Pijanowska made very small systems based on microfluidic, which allow to mix heat samples, biological samples, do enzymatic processes in order to determine urea in biological samples, just on spot somewhere far from civilization. In my group, we try to build up instrumentation for monitoring and imaging of blood perfusion and oxygenation in the human cortex, hoping that this will be someday very useful in every of the stages of treatment of patients suffering from post-traumatic brain injury or stroke. Starting already in the ambulance before the patient is transported to the clinics. There is an interesting device which we worked on in our institute during this pandemic situation. Many years ago, we developed, Professor Darowski, his group, we developed a device for independent ventilation of two lungs. Quite interesting. There are asymmetries in pathology between lungs, and this is quite useful concept. So you use one ventilator, and you can split the volumes between two lungs. But during this pandemic situation, we had a hope that this device can solve the problem of lack of ventilators and can be used to ventilate two patients using one ventilator. And we did some animal studies to validate this technology. Professor Ładyżyński works on something called voice diap system. This is a very simple smartphone application which allows you to say what you have on your plate and what you eat. And people with diabetes using this system can get information what should be the dose of insulin, which will be necessary to compensate this eating. In Poland, we have a structure called Committee of Biocybernetics and Biomedical Engineering of the Polish Academy of Sciences. The committee was established 50 years ago. So at the time when biomedical engineering was not really a buzzword known in the scientific community. So this was quite visionary task at that time. We gather specialists working in biomedical engineering from almost 20 different institutions all over the country. And I would like also to show you a few examples from the community, what people do in biomedical engineering labs in Poland. In University of Technology, AGH, we have group which works on biomaterials. They propose biomicro concretes which allow to repair bone defects very easily. The compound is biocompatible, bioactive, so it can be really used in solving real life problems. There is a very simple humanoid medical assistant, very interesting line of research, quite popular at the moment. So providing transportation within the clinic, measurements of various quantities from patients, delivering medications, but also providing a nice atmosphere for young patients, which is quite important. In Faculty of Mechatronics, this is my alma mater, a portable respiratory related impedance pneumography system is built, so which allows you to monitor respiratory system without putting mask on the mouth of the subject. In the group of Professor Iskander, there are activities related to corneal imaging based on speckle pattern analysis. Quite interesting idea with the use of OCT, quite innovative concept of how to get information on corneal function and structure. In University of Technology in Łódź, video endoscopy system allowing to monitor and diagnose problems in vocal cords. This concept is developed, and there's hope of commercialization of this. In Silesian University of Technology, they work on poly measurement system, which allows to monitor various signals from the body of the patient in order to assess the pain which is felt by the patient. So the question really is why these concepts do not convert easily into real products which are present on the market. And I tried to answer here this big question. Where are the challenges? First problem is that the cost of research in biomedical engineering is high. There are even figures showing this from some research, but we need phantoms, we need animal experiments, we need clinical validations phase. All these things are very expensive. We need standardization of the instruments. We need quality control, because this is a problem of safety of the
ponieważ bardzo często w zasadzie zachodzi
and what is really sad is that
pacjentów. Natomiast to, co jest naprawdę pesymistyczne, to to, że wielu naukowców wybiera specjalizacje, które są tańsze i zwykle powiązane z oprogramowaniem. Widzimy, jest to proces, który możemy zaobserwować również w Polsce. Wielu naukowców stwierdza, że mogą publikować lepsze publikacje w łatwiejszy sposób, korzystając na przykład z laptopa oraz kubka kawy, zamiast angażowania dużych grup w takim.
There is concept of valley of death, you know that, because there is lack of interest from point of view of scientists, or lack of financial resources, which appears very quickly after first feasibility study. But in my biomedical engineering, I would say there are two valleys, and I will discuss this later on. Mostly they are related to the assessment of the proposed technology by insurance companies. If the insurance companies do not pay for treatment, then the chance to earn money for the investors are limited. And then finally, there are huge ethical problems. We know that all the green wave around Europe press us to limit the experiments on animals.
which are really necessary for validation of biomedical engineering products. We would be happy to do experiments on unconscious patients, which are in principle not allowed by ethics. We are really afraid whether the algorithms can draw medical decisions, and many of the scientific activities at the moment are related to this topic. I will show you this later on. So the double valley problem is known from literature, and in biomedical engineering, this first valley is related to the situation that scientists give up. They already published this, so there is no reason to continue. But even providing that we find money to compensate this valley, we get another one, which is related to the phase where the product is already on the market, but the profit is very little because insurance companies don't pay for that. So in biomedical engineering, for biomedical engineering products, this process of commercialization is quite more complicated than for other products. We know that we have this whole wave of machine learning approaches, which may be useful for preclinical solutions, for computer-aided detection, diagnosis. These systems potentially can recommend the procedure of treatment. That's the idea, at least, or can give you early warnings of disease. For example, my watch gives you information about fibrillation of my heart, and this comes from analysis of the electrocardiography signal, which I can do using this very simple device. And there are two devices of this kind on the market already, which were actually tested or validated using huge number of patients around the world, and which uses the artificial intelligence approaches. The wave is huge. This is small advertisement from my institute. We have a journal called Biocybernetics, Biomedical Engineering, which is full of reports on how the artificial intelligence can solve this or that problem of medicine, and we invite you to publish in this journal. But then, if we talk about ethical problems, there are doubts of medical doctors, and these are quite serious doubts, that in all this environment of multiple devices, software tools, we can forget that the touch of medical doctor is something very important for the process of healing of the patient. And this is one of examples of this. Of these doubts. So this medical doctor said the most important innovation in medicine to come in the next 10 years is the power of the human hand. Just a touch of medical doctors. So the question is whether the patient can trust our algorithms drawing the most fundamental decisions of their lives, like, should I be treated with this or that procedure, which is extremely risky. And then I cannot move. May I ask for next slide? There is continuation of this, which is very general kind. I will not focus on that, but I would just only focus our attention on this book, which describes how the mathematical tools, simulation approaches can convert our lives, not only for good. Katie O'Neill is is the person who discussed that. And then I added one more problem. We discussed yesterday, we have to attract talents, the best candidates all over the world, who will come to our laboratories and do research in U13 countries. I'm afraid all these countries, in all these laboratories, we suffer from very low salaries of the scientists, which at the moment mean that most of these candidates just choose commercial partners. And then the big challenge, which I show here on the background of this paper, just published last year. The concept in the paper is to analyze whether the viruses present in our environment can be zoonotic, so they can be transferred from animals into humans. And probability can be assessed in this or that way, quite complicated, but this is only a list of first 26 of 50 such viruses, which scientists consider as viruses of high probability of spread from animals to humans, with unknown, in principle, consequences for health of humans. So what I say is the big challenge of biomedical for decision makers is to maintain scientific groups of complementary specializations in medical and biomedical engineering areas in order to be ready for the expected new diseases, new pandemics. Finally, very quickly, where are the unmet needs of medical doctors? We still don't have artificial, fully implantable heart. Big idea, big challenge, many solutions, many attempts. There were patients with implanted hearts maintained for many days or even months, but still the final product is not present on the market. Another big problem is artificial liver. So the mortality of this acute liver dysfunction is huge problem. And there are several solutions, as I show here, but last reviews show that these devices actually do not extend the period of life of patients in such a dramatic situation. So there is something to do for us. Maybe we should focus on higher resolution. So as you see, this is brain in 3 Tesla and 7 Tesla, if I remember correctly. So there is, so we can see many more detailed elements of the brain. But is this really a medical problem? Is this really useful in everyday medical diagnosis? Or we should focus rather on remote area radiology, very simple devices of low resolution, which will be available for far from hospital, far from power source. Or the most important problem of the medicine are just human errors. And there is idea of using checklists, which are again this experience comes from the technical sciences, from construction sites, from aircraft safety systems, which were developed by engineers. And so we engineers have already very good, very primitive, very well established tool, which is just a checklist, which is not accepted by medical community yet. So maybe this is really this is the biggest challenge of the medical theater at the moment. That's it. Thank you very much for your attention, and I hope this will be some basis for our discussion.
Thank you very much for very, very interesting and inspiring discussion. I think we can have time for a couple of questions from the audience. Otherwise, I have a long list, so we will start another seminar. But please, if you have any questions or comments to this presentation, Professor Liebert is ready to answer. There are not, so I have, can I start? I haven't noticed information about so important aspect of education of medical doctors as the simulation centers. These simulation centers that operate all around the world in all our medical universities are full of equipment that you presented and allow our students to start education in touch with artificial patient as early as the first year of the medical education, as I've discovered this year in Brno, for example. We also have at our universities these simulation centers. Can you comment on the limitations? Of course, you put some of them here, but can you comment on the limitations that are connected with limitations on technology that we have to stop and say, now we have to switch into hand-to-hand, face-to-face, and regular training on a regular patient in our hospitals? Because this can be one of the limitations you were talking about.
So being engineer, I should start with a very clear statement that I'm a fan of building up software tools which allow to model the situation, to provide systems which will allow the candidates to play with different parameters and receive outputs of the system which are similar at least to this which we expect in real life. And obviously I share this enthusiasm to build such models, but I also know how many aspects should be taken into account in order to make this model really useful. So a good example is this respiratory and cardiovascular system model, because we did plenty of experiments comparing the outputs with the outputs from animal model, from the situation in clinics. This allows to model different kinds of pathology and so on and so on. But to be honest, a very general model of human being is something what we dream of, and it's missing and it's very far away. Example of this is dialysis problem. In our institute we discussed for many years already problems of the flow of various chemical compounds through this membrane and things like that. And this is a huge area of interest of medical doctors, and still we can model only a few different processes. And so I would say, yes, wherever it's possible, this model should find a place in medical education system. But be careful, don't treat these models as something very well elaborated and very well reflecting the real clinical situation. I think the medical doctors with their experience and intuition will will never be at least in perspective of our lives. This still remains a big challenge.
Thank you very much for the comment. Any other questions or comments? If not, I would like to ask Professor Schroeder for his presentation, which will be focused again on challenges, but again what I said from different perspectives and different approaches. Okay, thank you very much for inviting me, and it's a great pleasure to be here. And I think it's also very special because I came from Dresden, and I think Roland and I, we can probably have a fight who had the shorter trip here to Breslau. For me, it was a 3 hours ride by car, and I think for you it was a 3-hour train ride. But I think everybody else who's not from Wrocław probably has a much longer visit here. So it's really great to be here. And yeah, I was given the task to present challenges and opportunities and directions in the development of bioengineering and focusing in particular on molecular and cellular issues. Now, I felt hopelessly overwhelmed, I must admit, with this title. So I said, what am I going to do? And I thought, I just tell you about the two stories that I found most impressive in the last, I don't know, 1 or 2 years that I came across, and I will tell you why I believe these things are so impressive. And then I've got a couple of slides telling you a little bit what I do in my research, which is a little bit connecting to some of these two issues. And so let's start. And that is number one. Who has heard of the hype topic liquid phase separation and condensates in molecular biology? Nobody, right? Raise your arm if you have. No, okay, nobody. And that is really amazing, the energy that is going into this topic. So the idea is the following. In molecular biology, everybody's concentrated on vesicles, where you have basically a membrane that is creating a microenvironment inside the membrane. And recently there were discoveries that influenced by external factors like temperature or pH value, you have more loose aggregates building that are not surrounded by a membrane, but rather they are compositions of RNA and protein, and they also do create microenvironments. And this cell paper is authored by a colleague of mine at Biotech in TU Dresden. And essentially, they basically created a connection between mutations in a certain protein that are known already to be connected to a neurodegeneration disease, ALS, and the formation of these condensates. And they can clearly show under the microscope that there is a connection with the mutations. The condensates form in a different manner than without the mutations. In a subsequent paper, they even found first drugs. And the hope is now that this whole topic of liquid phase separation and these kind of loose aggregates, these condensates, that this is an entirely new approach in molecular biology that will give completely new handle in drug discovery. And besides an astounding number of Cell and Nature papers that this colleague of mine is publishing, together with Tony Hyman from the Max Planck Institute in Dresden and a large group of scientists in Boston, a startup company was founded, although this is, well, I think really something different than the startups that one is usually used to. Bayer invested, I think, $100 million. And by now, I think this endeavor has received over $450 million, and they simply aggregating all scientific knowledge that exists around these condensates with a particular focus of on drug development. So also in my community, in bioinformatics, nobody has heard of these condensates. It's in molecular biology. It's a very, very hot topic. So stay tuned. And of course, one has to see what really comes out of it at the very end in terms of drug discovery. So that is hot topic number one that I find really very, very exciting. So hot topic number two, and now my hope is that a few more of you have heard about this, AlphaFold. So who has heard of AlphaFold? Just raise your arms. Okay, that's not so many. Well, that is for me personally in my bioinformatics research, really the thing I can say of my whole career that I found one of the biggest breakthroughs. And it's,
Yeah, I do a bit of advertising now for the people behind it. It's the solution of the structure prediction problem. So you have protein sequences in 1D, if you will, sequences of amino acids, and nature just spontaneously jumbles that up, folds that into determined 3D structures. And computationally, one was always thinking there must be rules behind it. There are forces, attractive and repulsive forces that drive this process. It must be possible to simulate them. But for many, many years, many teams around the world tried solving this, and they made progress, but they didn't really solve it until very recently, with the aid of a lot of computation, a lot of data, and in particular machine learning. A team from the DeepMind company solved this, and then hand in hand there was kind of the largest group working on this from David Baker in Seattle also solved this. There's another group in Boston that also did pioneering work. So essentially with machine learning on all of the data, this problem was solvable. And that's, for me, really a massive breakthrough, because I think it's the first time that there was ever an experiment done by mankind where really all available data was used. They really literally used all sequence data that is deposited in the sequence databases. And it was essentially a biological problem that was solved by a large team of mostly computer scientists, where I'm pretty sure that the vast majority of these computer scientists don't really understand much about biology. It was a small part of biology knowledge coupled with really massive computing and super data science expertise that solved this problem. And that makes it, for me, really novel and different to everything that I've seen before. Now, what are the consequences? And here now, first touch of my own work. In biology, or in bioinformatics, the key tool that everybody uses is BLAST. So you compare two sequences, you have a new sequence that you bumped into, you run it against a database, you find, oh, there are highly similar sequences in that database that are already known. They do something with cancer, so maybe my protein is also doing something with cancer, right? That's how a lot of molecular biology is operating for the past years. But the problem is that you need quite a decent sequence similarity in order to draw this inference. And we know that there are many examples where amino acid sequences have really more or less nothing in common at all with each other, and still they create the same 3D structure and have the same function that they perform. And here you see one beautiful example that we came across. Colleagues of mine are working on so-called diatoms. These are algae. They stick, for example, to the bottom of ships. They make the ship slower. So there's some practical need for working on this. And they want to understand how these algae adhere, you know, how the adhesion works. They identified a protein after they sequenced one of these algae. We put it into the AlphaFold structure prediction, predicted the 3D structure. And the question was now, what is this related to? Because by sequence, nothing at all was known about this protein, really nothing. So, and the structure that we predicted is the green one on the left. And then we ran a screen of this structure against all of the structures that were known until then, and that were solved with X-ray crystallography and that were deposited in the PDB. And as you can see, we picked up the middle one, the blue one. And on the right, you see a superposition. And if you haven't seen these images, it may look strange, but it pretty nicely fits on top of each other. And that is really remarkable. No sequence similarity whatsoever, but the structures fit more or less identically. And the strange thing is this blue structure is from a bacteria that is living under the ice sheets in in Antarctica, and it's an ice-binding protein. And out of the 8 most similar structures that we obtained from the PDB, 7 were from ice-binding proteins. And so that gives a handle on how evolution has worked here, and then how maybe horizontal gene transfer has taken place here. And this was only possible because we were able to predict the structure, and it gave us a handle to explain a little bit how maybe the adhesion is working, and give a handle to our colleagues how to do future research. Now, the second example in a similar vein is concerned with single-strand annealing proteins. So DNA winds around 11 copies of this protein, and it's a process that's still not fully understood. And the single-stranded DNA is then basically attached to the matching strand. And for 20 years there was a debate whether this protein, there exist 3 families that are completely independent, or whether it's one on sequence. Nothing was similar at all. And on the right-hand side, you see 3 columns, and in each column is representatives of the family members. And in 3D, they're really great similarities. So the blue part is really matching extremely nicely, and the blue part is exactly what is in the inside of that ring that you see on the left, or on a kind of helical structure. And so that is what needs to be conserved to create the ring structure. And what you have facing to the outside, that can vary and doesn't really matter between the different species. So with AlphaFold, we were able to settle this debate that existed in that community of my colleague for about 20 years. Okay, now quickly to my own work on structure-based drug repositioning. So drug repositioning is the idea that a drug can have multiple uses, that basically the side effects that we all complain about when we're taking drugs, that in certain circumstances they may be actually advantageous. And the reason is that a drug rarely binds just one target having one effect, but instead it's a situation where the drug is binding many targets, and the target can be, of course, be manipulated by many drugs. And in order, for the sake of time, let me speed out a little bit. Okay, so here you see that explained. So in the middle, you see the drug that we're interested in, and you see these different targets, and you see the binding between these individual drugs and the individual targets. But what you can see is actually, although these drugs are all different, in the part where they're binding, they fit perfectly to the target that we're interested in. So starting off from the dark orange drug, if we can find the light orange ones, they might be candidates that do maybe even a better job at the intended purpose of inhibiting that target. So we developed a software called PLIP. It systematically analyzes all protein structure data where drugs bind to targets. And instead of using the knowledge about the drug or the knowledge about the target, it just uses the knowledge of how the drug is interacting with the target. And it runs a screen against everything that is known about such target interactions, so against the whole of the PDB, and it figures out examples of similar interactions. And I'll quickly show you one particularly striking one. So this is actually a drug that is on the market. It's an antiviral against herpes, and in German, you can read there, it really says there, do not use this with chemotherapy, because there was already a drug repositioning attempt going on, trying to reposition this antiviral drug to turn into an anti-cancer drug, but that unfortunately failed, and the result is this warning over here on that package. So we analyzed how is this working in cancer, and we find that basically this ring here is sandwiched between those 2 rings. That's one pattern. Here are 2 hydrogen bonds, and so on. We code this in a vector, and this vector is then something that we can compare across the other databases. And essentially what we find here is these are the drugs that we picked up this way, and dark blue indicates similarity. So basically here are the drugs compared to all of the other drugs, and that is basically similar to what was known before. And the further away we get, to the more distant related drugs we get that nonetheless have the same pattern. And this is one that we picked up. It's amodiaquine. It's an anti-malarial. And you can see it has that same ring structure, the so-called pi stacking. Here's also a hydrogen bond. And so there are many similarities. And it turns out that in cancer cells, we could over and over repeat that amodiaquine was having actually a much better effect than the original drug called BVDU. And With the screening algorithm that we developed, we have repeated this in many different scenarios. So here, for example, one in autoimmune. So I'm quickly skipping through this for the sake of time. Here we have the same for a case in a South American infectious disease. And since we're talking also about technology transfer a lot, this has led to a spin-off company. So Joachim was a former PhD student who took the lead on that, and he spun off Farm AI. So it's a small startup company that had, of course, a hard time during Corona with 4 people now. And they're working together with TwoBind and NanoTemper. These are manufacturers of equipment to test drug target interaction. And Enemine is Europe's largest vendor of drugs. They are based in Kiev. So we're also in touch there. And it's actually fantastic to see how they manage in this difficult situation and keep the business going. So with that, thanks a lot for your attention to what to take away. Watch out for condensates. AlphaFold is really a big breakthrough that was driven by data, big computation, and machine learning. And with all of that data, machine learning, and so on, there is still a lot to do in drug discovery and how to make it efficient. And some of the algorithms that are presented can help in this way to make remarkable findings on new uses of existing drugs. Thanks.
Questions to the audience, if you have any comments or questions to our guests on this very interesting presentation. Yes, please. How do you actually measure chemical similarity?
I mean, what we're after, so basically there are different fingerprints. There are many. We typically use the Morgan fingerprint. Right, but our point is not that we make a selection based on this chemical similarity, right? We just obtain from our screen a bunch of drugs, and then our whole point to stress is that the drugs that we pick up, because they were found by interaction rather than by chemical similarity, that they really have very different scaffolds. And we typically use the Morgan fingerprint, but we have experimented also with many others, and of course, depending on which one you use, results look slightly different. Yeah.
Professor, yes.
Could you comment on this AlphaFold? So as I understood, you had the trouble to understand nature, and you collected a lot of data. And this allowed to predict or estimate how this nature will work for some particular situation. But the question is, do you have any reverse engineering idea? So to say how these processes look like, how they can be described by fundamental equations, based on this artificial intelligence process?
Yeah, that's actually a very good question. And Google and DeepMind, who basically solved this problem and got this big success, they say, of course, now everything's sorted out, no further questions. But of course, that's not true, because what we're really interested in is exactly what you say, is understanding. So how does nature do it? What are the forces? Describe this really in physical terms. And that problem is not solved at all. And the neural networks used for this machine learning task are so complex that also nobody really understands what is really going on inside. So we spent a really considerable amount of time and energy trying to do that, you know, to use explainable AI techniques to see what is happening inside these neural networks. You see that different topologies of proteins behave very different in these networks. You see the contacts that are formed. So you have a chain and it folds up, and then you have contacts, and how these contacts form, that you can see inside those neural networks. But of course, nobody, you know, could press a button and out comes you know, the physics force equation that would solve this once and for all. So you can predict protein structures, but the protein folding problem has not been solved at all and is as little understood as it has been before.
Thank you very much. Any other questions? If not, I will ask one more, if you allow. I would like to ask you to comment on the quality of data we use introducing to the system to get a proper solution. Because in my naive opinion, protein database bank offers quite well sorted out and standardized data that can be used to support your machine learning or any other artificial intelligence technology to be used to predict some other structures. However, if we go to less detailed or less well-structured data, how can we assure the final results to be, let's say, satisfactory?
So,
except of
excellent question, and I agree. So basically, in the PDB, we're like in the order of 200,000 protein structures, and that was good enough to predict the 200 million that are now available with this AlphaFold prediction. And I think a key why this was so successful is the high quality of protein structure data in the PDB. And for example, everybody is of course now interested to say, okay, what can we use this for? And this whole approach, can we apply it to other applications like drug discovery, which is so laborious, expensive, and so on. And I think that will be very, very difficult because the data situation is very different. For drug target interaction, it's not so standardized, it's not so high quality. You don't really understand what the authors
coming
have done.
from different.
And I had a wonderful discussion
yes.
with Roland on this. I'm sure you probably speak about that later, I guess, or. Okay, so we will see, but I guess we all agree that there is a big need to have such high quality standardized data also for many other areas outside PDB, and that will drive then applications of machine learning, and it's really a prerequisite for the success of these techniques.
Thank you very much. Once again, thank you very much for a very interesting presentation. And now I would like to start, in fact, the panel, I think, and we will ask maybe Professor Olga Stepankova, our guest from Prague, to start introduction and the beginning of the panel session. So I will convey microphone to Professor Adam Liebert.
Could we maybe start with the very general question of this bioinformatics? So continuing this idea of understanding the nature using artificial intelligence. What's your opinion where we are in this process? Yeah? So do we expect that understanding of this artificial intelligence process will lead to understanding of nature?
Well, that's a really, really tough question. And as a matter of fact, I believe that artificial intelligence at the moment is at the stage when it can help to change the laborious work of many persons into something what comes quick, and then comes the sprite, the hope that the people who will be around and interpreting the data will come with a sparkle that will bring the real new vision. Let me just mention some of the examples from our institute. Our institute has been established 5 years ago in order to support cybernetics, informatics, and robotics, and in order to bring new applications all around, not only in science, but also in industry. A big part of the institute is devoted to development of Industry 4 applications. And there is a small group of biomedical engineering that is trying to proceed in a similar way within the medical domain. And for example, we can build on some applications or solutions which we have been working on during last 15 years. Because, for example, there was designed a low-cost device for eye tracking in one of our former laboratories. And after those 14 or 15 years, this device is being applied in a completely different context in order to diagnose quickly and very early kids who will have some learning disabilities. And these can be applied in early stages already in nursery schools, while otherwise it is necessary to start with diagnostics through reading. So I believe that all these things which are being developed within the laboratories can slowly switch to the everyday life if we are trying our best to find out good applications. It was great to listen to all those fantastic results you have been talking about, and you have been talking about, because here this is the real interest of artificial intelligence, to get the inspiration for further development. For example, DeepMind has been designed in order to play chess, and finally, or Go. And finally, now it is solving real, true problems which are very, very important for understanding, for deep understanding of the principles. And this is just great, because artificial intelligence can't provide the understanding itself, but in good cooperation with the proper user, it brings the help. And the same with robotics. Another application which we are now working on is a smart home pet cat or pet robot that would be able to support people with dementia to live in their own homes. And what for? To help the persons, the diseased persons, but first of all, to free the hands of the carers to provide the hand which you were asking for, because the human hand is the best healing device. And this is exactly what I believe that artificial intelligence can do. It can free hands of persons to do the work for which we can be still considered to be the best.
Thank you very much. Thank you very much for your very human approach to the question of artificial intelligence and interaction of this with us. In the same lane, I hope Dr. Martin Rożanek will give us also a short presentation about his position and his possibility to use, let's use this simple word, use artificial intelligence for solving the problems of medical care of patients. I would like to say the word patients there, because although we are talking about the technology, we have to remember that at the end we are talking and thinking about solutions for patients. Martin, the floor is yours, and I hope the presentation is already
ready for, yes.
Looks like.
Yes, thank you very much.
Okay, thank you very much for the introduction. Good morning, ladies and gentlemen. Well, I have prepared the presentation, at least at the start, maybe more general, more about the biomedical biomedical engineering, because we have, I think, a little bit different situation in Czech Republic. Therefore, I would like to mention it, that our biomedical engineer is a regulated profession according to the national legislation. So it means that there is in effect, special law describing what such a program has to fulfill, that the graduates of that program are, let's say, approved or acknowledged as the biomedical engineers. And after that, it means that they can immediately start to work as a professional, non-medical, say, healthcare worker in hospital. That was the big effort of Czech Society for Biomedical Engineering. I have to say, before my involvement in the society. Therefore, it took quite long time, but now it means that it is a special position, and we have 4 universities in Czech Republic that have these programs, and the graduates are the biomedical engineers according to the law. These are in biggest cities, quite uniformly distributed along the Czech Republic. They are not the same, even if they have to fulfill the legislation, there are differences. So one of the universities, Brno is the example, is more focused to biochemistry. We have heard a lot about that. And another university is more focused on electronics and electrotechnology. Because I'm from Faculty of Biomedical Engineering, that I'm currently head of Department of Biomedical Technology. And we have the bachelor program, Biomedical Technician, approved by the Ministry of Health according to the law 96, and also Biomedical Engineer as a master program. Together takes 5 years, and the students typically, but it's, let's say, 50-50, start to work within the hospitals or start to work within the companies. What it allows us is like direct link, direct connection between the hospitals and between the universities. And originally, I guess it was considered the biomedical technician or biomedical engineer, the profession that was, I have used the bureaucratic. They were supposed to take care about the medical device safety testing and all these, let's say, legislative problematics. But nowadays, I see the shift that they are started to be the part of the professional teams within the hospitals. And they started to cooperate not only in the process of the healthcare about the patient, but also in the scientific parts of the care about the patient. And what I have prepared is like the idea what we should do, maybe with the medical data of the patients. Most of the devices, maybe almost all of them that are currently applied in hospitals have some possibility of export of the data, but it's almost without any further usage. So very often the medical devices are locked, so the extra payment is needed. And here's maybe the question, I can use some example. If you have a patient under anesthesia, there has to be the record from the anesthesia process. And what is the typical patient record? It's some typically A4 paper, which is handwritten by a nurse in 2022. So it's a little bit unbelievable, but it's still the typical medical record of the patient. And I think there is a huge possibility, huge potential to improve that, not only to improve the patient record itself, because of course we would limit the mistakes and improve the records, but as was said, there is a potential to work with these data and to maybe not improve, but maybe even improvement, but at least to optimize the healthcare in the hospitals. There are some examples of our graduates, our students that do work in hospital, and they do cooperate. They are part of the technical teams within, for example, the support in angiography in operation room within the surgery. We have, for example, programmers of cardiostimulators that are also directly in the operational room within the surgery. So these are, say, professional people that are directly involved in the healthcare. I see there a very big potential for the biomedical engineers, not only to take care about the medical devices, but they should be, I guess, directly part of the teams that do cooperate on the healthcare of the patient. So I've prepared one example at the end. It's the cooperation of our faculty. I'm involved there with one of the hospitals in Prague. It's, let's say, the data process of the patient with trauma brain injury. So we do some analysis of intracranial pressure and its behavior at the patient. Let's say the effect on the outcome of the patient and what can happen. And I've just mentioned citation of the article, which is a few months old in scientific reports, where are some our first results of analysis of the artifacts within these signals. And we spent maybe 1 year to work on the article, but we spent definitely more than 1 year to have agreement with the hospital to provide anonymized data to us. And it's something what I see that can very significantly limit the scientific work. The agreement, before it was approved by both sides, has to go, of course, through the lawyers of the hospital, of the university. Each of the institutes has special persons for GDPR rules, so there were another sides involved. And in effect, if I see the result as some scientific article, the biggest effort was to close the agreement between the hospital and between the university. And the smaller part of that was the scientific one. So that's, I think, it's what we should improve. To be careful, if I can say that the bureaucracy shouldn't limit the scientific part of that, because it limits also the development of the biomedical engineering. And one small thing I see is there may be some uniformation, or we should maybe think about how to unite the profession of biomedical engineers within the Europe. And my idea is that, for example, Germany has completely different system. So our graduates of biomedical engineering do not have the similar position in Germany, as I know, and different is in France. I guess in Poland will be also different. So there's probably another space how to improve maybe not only the scientific work, but also the healthcare in the area of biomedical engineering. Thank you very much for.
Thank you very much for this presentation and coming back to the earth and to the basic day in hospital and in the research. Questions and comments. I could see that there were some comments among the audience during the presentations. I think commenting that's the same problems we have here, and we will discuss in the afternoon sessions also. But any other questions? You know, 10 years ago, there was a paper, Insight paper in Harvard Business Review. The title of this paper was 10 Innovations That Will Transform Medicine. And among the list of 10 innovations, one of them was Patients Portal, which suggested that collection and access to data for individual will dramatically progress the medicine. We are 10 years later, and it seems that the problem, the big challenge has not been solved. And I can tell you, as a person involved in management of the university and the clinical hospitals, that we have different systems, had different systems 7 years ago in 7 different clinical hospitals, and they could not communicate. These days, after 5 years of very intensive work with collaboration with companies, we have 2 systems, and we hope that they will communicate within a few months, I think. So this is the scale of the problem. And thanking you very much for putting this into our session. I would like to go to other next speaker.
One more question, if you agree.
Sorry for that.
You say there is no access to the data in the instruments, and this is something what we suffer from in biomedical engineering community, because we want to collect this data, synchronize this data with other instruments which we build up, and so on. But what is really interesting are these portals or databases for patients. This data is not stored. I mean, and which does not allow to do any follow-up of the situation which happened already, especially for the ICU or the dramatic situations where the patient dies really due to the illness or due to some situation which happened on the ward. I mean, in 2021, we do not have enough hard disks to store this data, or what? Yeah. Where are the reasons?
Well, now I hope I not supposed to answer the question because it's really hard. Still, it's interesting because my experience is when the companies are supposed to do something around the information systems in the hospitals, they come and they almost first answer is, we can do whatever we want. We can do anything. It's up to you. And just as the time goes, the problems start to occur. The companies doesn't know how to solve it. So everything prolongs and it's difficult. And the example with the data processing from the final slide with the paper we already have is that we are, in that case, for example, almost unable to do some bigger research because the amount of patients is limited here. It's maybe 20, 30 patients per year. So that's why we have to switch to some multi-center studies, but these are difficult to realize, of course. And I think there's a lot of limitations that are very often underestimated. So like to synchronize all the effort. So, well, there's a lot of to improve, a lot of.
Thank you very much for your voice in this important discussion. Since we have half an hour still and 2 fantastic panelists, I would like to ask Professor Jak Willo to share with us his experience and probably interesting experience about Estonian approach to solve some of the problems of access to data, building a collaboration, and also to major questions of this panel. Jak, floor is yours.
Thank you very much for inviting. In the middle of summer, I got this invitation to Central European Technology Forum. I did not know what exactly to expect from this, but I'm trying to give a little bit of my take on this being Eastern European and back in my home country feelings. So, and I'm broadly talking about bioinformatics. As was said, I'm heading the computer science department, so I come from the IT side. It's important to understand where the people come from. So I was trained as applied mathematics and programming during the Soviet time in Tartu. Then I was lucky enough to go to Helsinki University, where I actually studied computer science and got involved in bioinformatics. And then I think one of the most important piece of my career was that I spent 3 years at the EMBL, European Bioinformatics Institute. Michael was wondering how come only 3 years, felt like long time. But this is important to go out and get the networks of people. And this is extremely important for every EU 13 country to get people go out, come back. I was invited back to work on the biobank establishment. It was funded by the private venture capital, and it was a separate foundation. The business side of that did not work out 2002, 2004, 5, but it was later handed to University of Tartu, and there is a major success story as biobank as a resource for research. And now looking more and more again towards commercialization options. And then from the university, I've been involved also with different company setups trying to help this technology transfer. I think some of the key take-home messages, I'd like to talk about these opportunities and challenges in biological and health data. We heard just about those. IT and AI solutions have many new applications, and it's really important to have a focus on people and talent. We all have this imposter syndrome, so being from this corner of the world, I'm not thinking as top researcher or nothing, but the ability to carry on is important. So networks, these European infrastructure networks, we are members of Elixir, the bioinformatics infrastructure. It basically covers all areas of biology. So all the databases for DNA, RNA, PDB, all of these data sources, some people need to establish, carry, quality control, monitor, develop tools. So we are involved in this process, as most countries now in Europe are. So being from the smallest country, one of the smallest countries in Europe, and I think smallest in Central Eastern Europe, so we carry this responsibility in here. And yes, we integrate with those tools, we contribute research, training, resources. But imagine the importance of these networks at the time of COVID, when every country started to do the sequencing of the viral sequences or counting, getting all the data. Without this global knowledge, we would be really in disastrous situation. So we need these collaborative networks to be able to cope with such situations. Then I move on to biobanking. So we work in Estonia with the genetic information of now 20% of adult population. So we have 200,000 individuals in the biobank. So effectively having a full sequence of about 3,000 people and genotyping certain marker positions for everybody, 200,000. But from this, we can infer partly also what is in between the haplotypes. And this gives the power to compare different groups, healthy, disease, figure out risk factors. And this resource is extremely important for research. So this slide, I hope that many of you have seen, but this is the cost of full sequencing coming down from the original 1 billion price tag to now well below 1,000 euro price tag for the full genome sequencing. But the genotyping part is even cheaper, 50 euros. One dinner worth, right? And the question is, can we put more value over the lifetime of that data back to the human health. So that's what is the goal, to have the genetic data of everybody and then provide services, some risk estimations, providing, preventing some of the wrong cure. So it can be used for cancers, although cancers are mutating all the time. So cancers are not really stable genetically. But for the diseases, we can have these, for common diseases, the risk factors. For rare diseases, there are usually some genetic variants, very specific. And one where we see the promise is the pharmacogenetic. So we talk about the drug repositioning, but in our bodies, the enzymes are breaking up the drugs. So if I do not produce the enzyme, the drug accumulates. If I produce too much of the enzyme, there is no time for drug to interact with my body. So these kind of things can be also detected from the genetic sequence of humans. And yes, from research, all this health data, billing data, we have all online prescription system now in Estonia, potential imaging that is tough. So this data adds value to the genetic data in the biobank. Imagine now having the standard biomedical or health research question, like we figure out that if you do this and this, then you have this outcome. So can you repeat my study in Czechia, in Poland, in Wrocław, in Warsaw? Everybody would go to the database, start to extract data, start to do some scripts on their own because data looks different in every place. It doesn't scale up, right? So one of the approaches where we are engaging now is this Odyssey bottom-up initiative to come up with a common data model, OMOP common data model, where the idea is that you at home map data to this common data model. The other country maps to the same data model. You don't need to share the data because that is ethically, GDPR-wise, very complex. But once you have the script on one database, you can apply the same script on the other database. So we work with these common data formats, or actually already the database structure, where the studies can be repeated from one place to the other, and the results aggregated, compared, looked for differences, for reasons of the differences in the medical systems, in the coding systems, you say what. And having this power of health data, genetic data, what we are really struggling at the moment or completing is to build a national system where the medical doctors can access the genetic information from specific patient through the decision support system on the genetic component, and get the warnings to the medical doctor or patient, the risk factors and pharmacogenetic predictions. AI, where you know perhaps the background that big breakthrough in AI was in image analysis. The deep neural networks really broke the image analysis challenges, and this has been now, we have collaborated with the PerkinElmer to analyze microscope images to the AI on imaging. I'm not going into details of all the research aspects, but there is one important key. What I want to deliver is these knowledges are transferable from one situation to the other. So learnings from the chess and Go to the protein sequence, protein structure analysis, image analysis, counting certain types of cells, to self-driving autonomous cars. So focus on these technologies have a lot of applications, not in that one specific context, but also in the other. So we have people coming from the computational neuroscience background, deep neural networks, bioinformatics, who have also contributed the alumni and research to cars on the street. So we are collaborating with the Estonian startup Bolt, that is also luckily in here available. IT is important. This is our institute student numbers over the last dozen or so years. University has lost 1/3 of population, so we are the only discipline that has been growing strongly. Estonia lost birth rates, like 45% of birth rates in beginning of 1990s. But IT is important. We can say that it's only software on my laptop, but actually it's the underlying all the technologies that are used in biomedical engineering, bioinformatics, health data analysis. In every aspect, we need software components. So now we have 1,500 students, and on the teaching side, over half of our teaching staff, research staff is international. So this brain drain that we have lost to the West, we also have to reverse, bring back people, bring in new people, because we have not been able to produce all the PhDs, all the researchers ourselves. So in that sense, we are well connected, and the research is going strong. And because of this growth, this is our new building in the Tartu city center, this Delta, and yeah, infrastructure, buildings are also important because we are competing with a local startup scene. We need all the hardware for compute. So we have our own computer clusters and systems, of course, for AI, but we are also collaborating with a Nordic Lumi supercomputer that is now Europe's fastest, still being launched on finding the final bugs in the systems, but in Europe, the fastest, worldwide third fastest. So it's important to have also these resources for all the AI that we want to apply. And with this, I bring back the key messages: focus on people. Many of these technologies are transferable from one situation to the other. It's really important. It's not only a single bet on one aspect, but it has many different ways of returns. And we need to have people back. So with this, I'd like to finish at the moment. Thank you.
Thank you very much for your presentation. Questions or comments to the very interesting presentation? Yes, please.
Smart people, they tend to do their PhDs and they find much more interesting work in English.
Oh, I have had so many different questions on these topics. Why do you need to even have the bachelor degree if you can do scripting without any degree, or the coding schools, no degree needed? So I have always said that we need PhDs, master education. The master education has been growing. PhDs, we have not produced far enough even to have our own university catered for. So, and of course, out of the alumni, half or more work in the industry. That's how it should be, right? But we should, as countries, we should focus so much more on PhD education to have the next generation of teachers, technology developers, etc. We should be proud when they do that. But our governments are a little bit lazy to understand that we need more of those PhDs. On the other hand, a bit of advertisement, I think we are now paying better salaries to PhD students than Italy would, for example. So it's not that we are not paying to young researchers. Yes, the local jobs at Bolt or Wise or other Pipedrive, other unicorns is still higher, but that's life. Yeah, as a researcher, I can't say that we are paid little, but the comparison to the local industry is, of course, still big. But we are catching up very, very rapidly to Western Europe.
Thank you very much. I would, before I give floor to Professor Roland, there is another question. Thank you.
Can use the genomic data in the treatment of patients.
No, I have been managing the project over the last 4 years. We are at the last month's half a year where we can really provide this repository for genetic information and bring out the data in the compute environment to apply the software as a medical device. What we are lacking is that we have all the research knowledge, we have the R scripts, we know what to calculate, but we are lacking this certification of medical devices, software as medical devices, notified bodies, and this kind of commercialization step at the moment. Who takes the responsibility? We have the research knowledge, but we lack this one final step. And Estonian government, basically now there is a call out. So who are the interested companies in Europe to provide such pieces of software? So technically we are almost there to launch. The other challenge is that the research data in the biobank has been analyzed by the research-oriented chips, Illumina chips, for research use only. And transferring that to the medical system is a bit of a challenge. They have been following the ISO processes, etc. They can be audited, but the hurdles are really the legal hurdle, not anymore the technical so much. So we are so close, yet so far, kind of.
So it can be done as the medical experiment.
So there have been medical experiments carried out for breast cancer screening. So if you have high breast cancer risk profile over thousands of markers, then you can start inviting people earlier to screening. So this is one first use case, and for the pharmacogenetic use, we have kind of we know what to look for. But the implementation as medical device is a little bit challenging at the moment. But we are very, very close to full-scale launch.
Thank you very much. I quite recently listened to the story of Estonia's success, and I know that there are only 2 procedures that can't be done participating face-to-face. Otherwise, you have e-services. So this is marriage and selling property. All other services are offered as e-services, as somebody told us. So congratulations for this.
But the property can now be bought online.
Really?
Yes,
oh, so it changed
I have been buying that with my daughters. Yeah. Because over the COVID time, all the identification, everything is there. Electronic signatures are there.
So that's unbelievable achievement. Thank you very much for your presentation. And we have the last panelist. So I would like Professor Roland Vogelmuth for his reflections, his comments on the field.
Yeah, dear Chair, dear ladies and gentlemen, it is a great pleasure for me to be here with all of you today at this Central European Technology Forum in the wonderful city of Wrocław. And I would like to thank the organizers for inviting me. I enjoyed very much here the presentations and discussions of these sections and also yesterday. So let me first share with you some relevant technology trends from my point of view before discussing most promising research directions, how to improve EU financial instruments and their implementation schemes, how to broaden and strengthen international cooperation, how to overcome present-day barriers hindering cooperation with European industries. So I should move to the next slide. Sorry, no, still with the technology trends. So We all know biotechnology has made beneficial contributions to mankind for thousands of years, and the COVID crisis has demonstrated clearly the power of biotechnology as a key enabling technology. Biotechnology is not only essential for the fast development and manufacturing of novel diagnostics. We think of PCR, omics, enzymes, and therapeutics like antivirals, antibodies, enzymes, messenger RNA, but also for a range of novel biomaterials, biopolymers, specialty chemicals, which are also of importance for medical devices and different auxiliaries in healthcare. Biotechnology, as we have seen, is also a key enabling technology for the fast development and manufacturing of messenger RNA vaccines, where the companies manufacturing the world supply can't find enough people in the time they are building the new factories. The development of in vitro transcribed messenger RNA for protein therapy is a very promising and attractive ongoing area, for example, for cancer therapy. We have heard yesterday about the human-centric approach from Peter Dröll, and it is in this direction that it is exciting to see that fast instrumental analytical techniques are moving to the point of care and to the point of medical intervention, for example, directly in surgery, if we think of the eye knife. Next slide. What are the most promising and thus requiring priority European funding research directions from my personal point of view? The elucidation of the molecular mechanisms in all 4 domains, and I emphasize the 4 domains of a true molecular biology, because classically, of course, only DNA, RNA, proteins are considered as the key domains in the central dogma of molecular biology. But as we know, the small molecular domain, the 4th domain, the metabolites, are also important because they can influence expression of oncogenes or expression of tumor suppressor genes. So these molecular mechanisms, which are underlying human diseases, is a key success factor for developing effective and precise diagnostics and therapeutics, for example, in the areas of infectious diseases, inborn errors of metabolisms, which we know since the times for more than 100 years of Sir Archibald Garrett, who was the pioneer of analyzing just the human urine of a patient, and from the changed color, he concluded that this is part of the chemical individuality. So he identified it correctly as a genetic disease long before the structure of the DNA has been found. So inborn errors of metabolisms, which we now have know more than 700, are an interesting area for these molecular mechanisms. But also cardiovascular diseases, cancer, and neurodegenerative diseases. Next, genetically defined patient cohorts are of great value for the discovery of new connections between diseases and metabolic pathway changes. For example, cancer cell metabolic reprogramming. We have heard a lot in past presentations yesterday and today about the importance of standardization. So standardization of sequence, structure, function, reaction relationships, and their storage enables facilitated knowledge transfer across discipline boundaries. For example, from somebody working in microbiology towards medicine, looking at an infectious disease in the microbiome caused by that exactly same microorganism. The development of fast and meaningful tools for precision diagnostics is a prerequisite for the development of therapeutics and for medical decision making. Effective and precise therapeutics, which are manufactured in Europe, will make Europe more independent of interrupted global supply chains and overcome the non-availability of essential medicines in Europe, which is, according to a medical doctor who does the statistics, enormous, and that's improving the day-to-day situation in European hospitals. So moving back production, because there was a tendency from pharmaceutical companies to outsource production because the return of investment, for example, for antibiotics, was not good enough, so it was outsourced. But when, of course, the production company somewhere on the globe goes bankrupt or stops production, the pharmaceutical is no longer available worldwide. So moving back production from Asia to Europe is not only a technology opportunity, because one can also improve the ways how these pharmaceuticals can be manufactured, but it is an opportunity for a coordinated European approach involving science, industry, government, and also society, because society has perhaps to accept that antibiotics have life-saving antibiotics need a higher price. And this is the perfect European approach, because no single European country can solve this alone. How to improve the EU financial instruments and their implementation schemes? So from my point of view, EU financial instruments are already working very well, from fundamental research through theme-oriented research to public-private partnerships. Although times of high inflation affect us all, increased support of students, master students, doctoral students, and postdoctoral researchers would be beneficial for natural sciences and medicine. Facilitating scientific curiosity and exploratory research for early stage researchers would also be attractive. In order to make the continuous supply of essential medicines to the European healthcare systems more robust, financial instruments for revitalizing manufacturing in Europe of critical pharmaceuticals, which are system relevant for the health of European people, would be good. How to broaden and strengthen international cooperation in this area? We have heard yesterday that trust is a key element of cooperation, and therefore it is essential to aim at consensus finding in the scientific community regarding the experimental measurements of meaningful and robust data. And we have heard already in the talk from Michael Schroeder pointing out the AlphaFold, the importance of the structure data, the about 200,000 protein structures in protein data bank. That was a key prerequisite for developing this major breakthrough. And I'm involved, I don't have time to go into all the details, but I'm involved in the Global Strender Commission, which has worked out guidelines for describing the protein function, for example, in the 7 enzyme classes of the EC classes globally. And because it's a prerequisite that the global scientific community finds a consensus how to publish that, in what format. So to make it retrievable by machine learning and computer scientists to bridge that. And that was quite a lengthy process, but we have now achieved these guidelines, which are recommended by more than 50 editors-in-chief of the major life science journals globally. And in addition, we have launched at the European Bioinformatics Institute in Cambridge the Strenda database, which will become the functional equivalent of the protein database PDB. It will take years. It will not be finished in 1 year. And of course, because the deposition of the function data in the Strenda DB, we have not made mandatory yet, because we thought we rather want to convince the global scientific community that it is worthwhile to go an extra mile for the individual, because of course we are all busy with things. But if, let's say, the global scientific community would each person publishing one of these more than 50,000 enzyme papers per year, do the extra step of depositing this in a standardized way, this could enable completely new areas. And I think I have to stop here, make it faster. And I move on to the next point. This is equally important for the standardized description of biodata. For example, what I mentioned, the Strenda database. This is a prerequisite for building global databases, and databases should be designed from the beginning for international cooperation, where data formats are interoperable on a global level, create value for both Europe and the global scientific community. And last but not least, bringing together international experts in a conference like this here or monthly webinars inspires new cooperations. So next slide, how to overcome barriers. First and foremost, it is the scientific ambition for attractive and excellent research. The communication between industry and academic institutions is important for both sides. And a clearly defined and unique research focus, which stands out internationally and is attractive to Europe's leading industry, is a good starting point for possible cooperations. And in order to smoothly overcome barriers that hinder cooperation, it is important to pay sufficient attention to differences in goals and expectations, and to resolve administrative and legal incompatibilities between research institutions and Europe's leading industries. And to conclude, I see I have taken up too much of time. Sorry, Chair. I just end by the last slide. Diseases do not recognize boundaries of countries or protocol systems, but diseases recognize people. Thank you very much.
Thank you very much for your presentation. I want to keep this slide because I think it can be a fantastic summary of our panel. So before we leave, I want to thank very much to all panelists, and I hope you will answer the questions during coffee and tea break, and thank you for that. We want to take your time for coffee break. Once again, please join me in applauding our speakers and panelists for having for being here today with us and sharing important factors. Only one word from each of you before we leave. One word that you think is quite key for development in the direction that we discussed today. One word.
The data resources.
Fantastic, Professor. Sorry?
Ethics.
Ethics. Go with that.
Smart data standardization and data sharing across Europe.
Fantastic.
High quality data.
Personalization.
Thank you.
precision medicine.
Thank you very much. And I would add human resources and patience, because this is something that we have at the end of this chain. Once again, thank you very much. Enjoy the rest of the conference, and thank you for coming.